Thank you so much for the introduction, and thank you to the organizers for giving me the opportunity to share my current and future research. My overall research interest is in the biology of sexual reproduction. This is a fascinating process used by many species to generate the next generation. That includes us humans. This is an important process because without it, many species, including us, would not exist. And it is fascinating because of the complexity and diversity it takes to make this process work. Throughout my PhD and postdoctoral work, I've been interested in how sexual reproduction is regulated and impacts physiology and health. I've been studying these questions across scales, from subcellular structures to cell types to tissue and organ and the whole organism. I've been studying these questions across time, from the half-life of a protein in hours to the lifetime of an animal in years. During my PhD at UC Berkeley, my work focused on how is sexual reproduction regulated. I study how chromosomes are segregated in meiosis using genetic and cell biology tools such as light cell microscopy. During my postdoctoral work at Stanford, my current research aims to understand how sexual reproduction impacts physiology and health. And I've been approaching this study from the angles of cell types and tissue and organ, and importantly, during the lifetime of an animal. In other words, during aging. Aging is associated with progressive tissue deterioration, frailty, and disease. And it is a process that can be influenced by life experience. One of such life experiences is sexual interaction, also known as mating. This is a behavior crucial to sexual reproduction, and it occurs in most individuals at some point in their lives. And yet, compared to other external factors, such as diet and exercise, we know very little about how sexual history is related to aging and age-related disease. In fact, most of our understanding of how sexual interaction affects aging comes from two invertebrate species, C. elegans and Drosophila. And these studies have shown that mating can regulate the physiology and lifespan of an animal. But what's not known is what is the impact of sexual interaction on vertebrate aging, especially on vertebrate-specific organs. How are the invertebrate mechanisms adapted to accommodate vertebrate-specific features, and are there new mechanisms? Currently, these questions are challenging to answer because of the long lifespans of existing vertebrate organ system, for example, mice, and they live around 2 to 3 years. And to address this challenge, I've developed the African tortoise killifish as a new vertebrate model to study how sexual interaction impacts organ aging. The killifish are native to Africa. They live in these annual ponds that will completely dry out during the dry season. And this extreme habitat led the killifish to evolve a compressed lifespan of 4 to 6 months, and that's about a fifth of the mouse lifespan. And a killifish is also a great model system for my question because it has vertebrate-specific organs and systems important for regulating aging, such as adaptive immunity. And lastly, for the past 20 years, the killifish field has developed a panel of husbandry, gene editing, and molecular tools, and I've contributed to 3 of them. As a field now, we're well positioned to answer mechanistic questions. So now to mate, the killifish displays specific behaviors. The male would swim around the females, press on her belly to push the eggs out. At the same time, he would release sperm to inseminate the eggs. So when the killifish mate, there's no internal fluid exchange between the mating partners. The embryos are fertilized and develop externally without parental care. So using the killifish, my specific question is how does sexual interaction or mating affect organ aging in killifish males and females? To answer this question, I compare killifish males and females that are either unmated or mated lifelong. I surveyed a panel of somatic organs, including the brain, gut, kidney, and the liver. Interestingly, the liver is a vertebrate-specific organ and is known to be important for reproduction. Therefore, I decided to first focus on the liver and use it as an archetype to understand how mating may regulate organ aging in a cellular and tissue level. To characterize the mating effect on liver aging, I use a combination of approaches, including single-cell RNA sequencing, in situ staining, lipidomics, and building machine learning models. And together, these approaches have allowed me to characterize the mating effect in an unbiased way, and in several cases, in a cell-type specific manner. So using these methods, I was excited to find that mating affects immune cell balance in female livers during aging. Here I'm plotting the proportion of cells that are macrophages in female livers. On the x-axis are the different conditions that we're trying to compare, and each dot here is an animal. As we can see here, with age, there's an increase in macrophages level in both the unmated and mated females. And this is consistent with the observation that aging increases immune cell infiltration in other animals. Now, what's interesting is when we compare all mated to the all unmated females. There are fewer macrophages in all mated females. And this suggests that mating dampens macrophages levels at all age. And importantly, this dampening effect does not happen in males, and suggesting that this is a regulated process. Now, in addition to immune cell infiltration, I also observe that hepatocyte inflammation is lower in all mated females. Using machine learning models, I found that these hepatocytes have a younger transcriptomic age. And this is particularly exciting to us because mating has not been previously implicated in modulating immune cell balance, inflammation, and transcriptomic rejuvenation in the liver. Now, how about liver functions, such as metabolism? Using lipidomics, I was excited to find that mating affects lipid metabolism in all female livers. Here's a heat map for lipid abundance. The color orange here means a higher level. Each row is a lipid species, and each column is an animal grouped by conditions. As we can see here, at the OH, mating upregulates specific lipids, such as these ones that belong to the class called phosphatidylserine. And importantly, these lipids are not different by mating status in the young age. And this suggests that mating affects lipid metabolism in an age-dependent manner. Now, to summarize, what I've discovered is that sexual interaction affects liver aging in at least two aspects: metabolism and inflammation. And more broadly, using bulk RNA sequencing, I also observed that these two aspects are altered by mating for the female brain and female gut. And this suggests that mating may modulate organ aging in a more global manner. And together, our work provides the first evidence that mating reshapes the physiological context during aging in a short-lived vertebrate. Now, moving forward, I'm very excited to expand on what we know in three main ways. First, I want to dissect the molecular mechanism by which mating regulates liver aging. And our current hypothesis is that liver-ovary communication may play a key role. And I have constructed several mutants to test this idea. And secondly, I want to understand how mating can affect the cell biology of lipid metabolism. Given that lipid metabolism requires subcellular organelles, I want to know, how does mating affect organelle dynamics, functions, and coordination? And lastly, I want to understand how mating may affect the resilience of an animal against stress and disease. And for that, I have already developed 3 systems, including drug overdose, liver regeneration, and overfeeding. My future lab will test how mated and unmated killifish respond to this system while developing other disease models. In the long term, I envision that the work from the Chen lab will help establish a vertebrate paradigm to guide our thinking on how human sexual history may relate to aging and disease. And I think this work will have implications for personalized medicine, disease treatment, and the design of new interventions to counter aging and age-related disease. With that, I would love to thank my postdoctoral advisor, Anne Brunet, for her tremendous support of this project, the entire Brunet Lab for being a great place to do science, my past and current students, mentors and collaborators, and funding. And thank you very much.